Endocannabinoids and the Endocrine System in Health and Disease. Cecilia J. Hillard. 1. Author information. Copyright and license information. PMCID, PMC 6813821 NIMSHID, NIMS 1054664 PMID, 26408166. The publisher's version of this article is available at hand PEXP Pharmacol. Abstract. Some of the earliest reports of the effects of cannabis consumption on humans were related to endocrine system changes. In this review, the effects of cannabinoids and the role of the CB1 cannabinoid receptor in the regulation of the following endocrine systems are discussed: the hypothalamic pituitary gonadal axis, prolactin and oxytocin, thyroid hormone and growth hormone, and the hypothalamic pituitary adrenal axis. Preclinical and human study results are presented. Keywords: gonadotropin, gonadotropin-releasing hormone, testosterone, prolactin, oxytocin, growth hormone, thyroid hormone, HPA axis, corticosterone. 1. Introduction. Endocannabinoid signaling, ECS, plays a wide variety of modulatory roles throughout the central nervous system, CNS. The endocannabinoid system consists of two G protein-coupled receptors, CB1 receptor, CB1R, and CB2 receptor, CB2R, the vanilloid subtype of transient potential receptor and members of the peroxisome proliferator activated receptor family. 2 endocannabinoid, ECB, ligands have been identified, N-arachidinoylethanolamine, AEA, and 2-arachidinolglycerol, 2-AG. Both are synthesized from phospholipid precursors in an on-demand manner and are metabolized by hydrolysis. AEA is hydrolyzed by fatty acid amide hydrolase, FAAH, while 2-AG is hydrolyzed by monoacylglycerol lipase, MGL, and by alpha-beta hydrolase 6, Mars ETAL, 2010. Both AEA and 2-AG are also substrates for cyclooxygenase 2, which converts them to ethanolamide and glycerol-substituted prostaglandins, respectively. Hermansen ETAL, 2014. Within the CNS, ECS mediates activity-dependent, retrograde signaling in many brain regions, including the hippocampus, prefrontal cortex, amygdala, and cerebellum. Freund ETAL, 2003. In most cases, 2-AG is mobilized in postsynaptic neurons by receptors that activate phospholipase C, PLC, including the metabotropic glutamate family of receptors. The diacylglycerol, DAG, that is produced is further metabolized by DAG lipase to monoacylglycerol, including 2-AG. 2-AG acts on presynaptic CB1Rs to inhibit neurotransmitter release, through inhibition of the opening of voltage-operated calcium channels. The CB1R is also present outside the CNS, including adipose tissue, liver, and the adrenal gland. The CB1R in adipose and liver promotes the storage of fat and reduces fat utilization. Silvestri ETAL, 2011. There is little known about the sources of the ECBS that innervate the non-CNS CB1R. However, the ECBS are present in the circulation and recent data indicate that the circulating concentrations of 2-AG are nearly 4 times higher at noon than at 4 a.m. in healthy humans. Hanlon ETAL, 2014, leading to the hypothesis that circulating ECBS activate these receptors and thereby coordinate adipose and liver function with caloric intake. Formulations of the cannabis plant have been used by humans for thousands of years for the treatment of a variety of conditions, including pain and spasticity. Kumar ETAL, 2001. Delta-9 tetrahydrocannabinol, THC, is a direct agonist of the CB receptors and is responsible for these medicinal effects as well as the feeling of euphoria or high that is sought by those using cannabis recreationally. There are many other chemicals in the plant that also have beneficial effects, but whose mechanisms are not as well understood. Devinsky et al. 2014. CB1Rs are present in the hypothalamus at relatively low density compared to other brain regions. Herkenham et al. 1991. However, it is argued that this population of cannabinoid receptors is highly active, given the broad range of endocrine effects of the cannabinoids. Fernandez-Ruiz et al. 1997. Within the hypothalamus, the CB1R protein is heterogeneously distributed. Whitman et al. 2007. CB1Rs are present on both symmetrical and non-symmetrical synapses, and most immunoreactivity is in preterminal and terminal portions of axons. There is sparse CB1R distribution within the suprachiasmatic and lateral mammillary nuclei, but other regions of the hypothalamus express significant amounts of CB1R. Hypothalamic CB1R density differs between male and female rodents. Rodriguez de Fonseca et al. 1994, and this likely reflects important sex-related endocrine differences as well as differences in cannabinoid effects between male and female animals and humans. Kraft et al. 2012. CB1R mRNA has been identified in the external zone of the median eminence. Whitman et al. 2007, Herkenham et al. 1991, and CB1Rs are expressed at low levels in the intermediate and anterior lobes of the pituitary gland. Pegato et al. 2001. Two cannabinoid interactions with the hypothalamic pituitary gonadal (HPG) axis. The first step in the regulation of the HPG axis involves the peptide hormone, gonadotropin-releasing hormone, GnRH, which is produced by neurons in the preoptic area of the hypothalamus. GnRH secretion is pulsatile and it affects the release of two pituitary hormones through receptors in the anterior pituitary. Low-frequency GnRH pulses induce the release of follicle-stimulating hormone, FSH, while high-frequency pulses induce luteinizing hormone, LH, release. In males, the frequency of GnRH release is constant, while in females, frequency increases significantly at the time of ovulation, resulting in a surge of LH. FSH and LH regulate follicular growth, ovulation, and maintenance of the corpus luteum in females and spermatogenesis in males. CB1R activation inhibits the release of GnRH through effects in the hypothalamus in male rats. Studies in isolated hypothalamic tissue demonstrated THC-induced suppression of simulated but not basal GnRH release. Retieri et al. 1990, and inhibition of pulsatile GnRH release by other CB1R agonists. Gammon et al. 2005. Mediobasal GnRH was increased by intracerebroventricular (ICV) THC treatment. Data consistent with a reduction in GnRH release in the pituitary. Wenger et al. 1987. However, another study demonstrated reduced concentrations of GnRH in the preoptic area and mediobasal hypothalamus following in vivo THC treatment. Kumar and Chen, 1983. It is possible that the discrepancy between these two studies is the result of different stimulus durations. Cells adjacent to GnRH secreting cells express CB1R mRNA. Gammon et al. 2005, and a subset of neurons forming symmetrical synapses with GnRH neurons express CB1R protein. Farkas et al. 2010. CB1R activation inhibits gamma-aminobutyric acid, GABA, release onto GnRH neurons. Glanowska and Moenter, 2011. Farkas et al. 2010. Data in agreement with the predominant effect of CB1Rs being suppression of neurotransmitter release. Freund et al. 2003. Reduced release of GABA is associated with increased excitatory drive. so these data seem to contradict the findings described above that CB1R agonists inhibit GnRH release. However, GABA can depolarize GnRH neurons under some circumstances, Herbison and Moenter, 2011, and thus, inhibition of GABA release could paradoxically decrease GnRH neuronal activation. Alternative mechanisms have been suggested, for example AEA-induced inhibition of GnRH release evoked by NMDA in mediobasal hypothalamic fragments was blocked by both a CB1R antagonist and bicucoline, suggesting an increase in GABA release. Fernandez-Solari et al., 2004. Other data suggest that THC acutely suppresses norepinephrine stimulation of GnRH release, Steger et al., 1990, Murphy et al., 1990. Chronic treatment of male mice with bang, a cannabis preparation, results in reduced expression of receptors for GnRH in the pituitary, Banerjee et al., 2011. Thus, while the effect of activation is consistently depression of GnRH release, currently available evidence suggests that multiple mechanisms are involved. Given the differences in patterns of release of GnRH between male and female, it is not surprising that sex steroid status profoundly affects CB1R regulation of GnRH release, Scordicati et al., 2004. In particular, AEA had no effect on GnRH release in overectomized, OVX, rats and increased GnRH release in hypothalamic tissues from OVX rats in which estrogen is replaced, Scordicati et al., 2004. In agreement with findings obtained in experiments with other systems, Kraft et al., 2012. It seems that estradiol is an important contributor to differences in response to CB1R activation. An earlier study also found opposite effects of in vivo THC treatment on hypothalamic GnRH in male and OVX female rats, Kumar and Chen, 19.